Let's take a look back at the roots of Codetta Bio with co-founder Mike Ramsey. What was the catalyst that led to starting Codetta Bio?
Yeah, well, that's an interesting question. I started my first company, Caliper Technology, became Caliper Life Sciences in mid 1990s, and that was a great experience. I found it very exciting, and um, sort of moving on out. I mean, I'm an applied scientist, right? So applied scientists, right, I think should look to address unmet needs. In science and technology. And so, particularly when I moved to UNC in 2004 from Oak Ridge National Laboratory, the projects I worked on, I wanted to work on projects that were challenging. I was a tenured professor at that point, and projects that addressed unmet needs. So, since we're an applied sort of science and technology developers, I always thought of a product at the end of the road, right? That we were going to do basic research and then development, and it should lead to a product, and naturally then a company that could commercialize that product. So that's how I've thought about my research for the last 20, 25 years in particular. So it was sort of natural. We had this BAA from DARPA that came out that basically wanted to address state-of-the-art technologies for making protein measurements and nucleic acid measurements, genetic assays. And we came up with some great ideas. Hamp Henley, who was in my lab at UNC, he certainly played a key role in creating the technology that Codetta is using, licensing from UNC today. And, those ideas, it was naturally for me to think about forming a company to commercialize those. Even though it may have been 5 or 10 years out, and which it actually was, right? We started working on the technology in 2011. So we've had a few years of development.
Was there a specific problem or a need in the market that drove the founding team to develop the Codetta technology?
Yeah, so this DARPA BAA that came out circa 2012, right, it addressed needs for diagnostics in particular. So they wanted, say, state-of-the-art capabilities in protein assays as well as genetic assays, nucleic acid assays. So they sort of built a roadmap for needs in the diagnostics industry, and so that was sort of they had done the research of needs in the diagnostics industry. So, that was the basis for development to addressing those needs with Codetta technology. Yes. So, sort of the foundations there of DARPA needs was, state of the art in terms of detection capability. They also wanted high multiplex capability, and also the ability to change the menu very rapidly. So reconfiguration of a menu to address emerging needs.
What was the original vision for Codetta, and how has it evolved?
Yeah, so the early vision of the company was, again, largely driven by this DARPA project, which we eventually were awarded $20 million to develop the technology, the Codetta technology. Yeah, the early desires of DARPA was a sample to answer platform to address diagnostics, right? So essentially in vitro diagnostic type of measurements, both from a protein sort of basis, doing genetic assays and doing protein assays. So a single platform that had the ability to address both of those needs. As we started to sort of market or test the needs, commercial needs, we found that people were more interested in an RUO platform that was high throughput in nature. So that sort of changed our our thinking from the DARPA program, which was sort of a small instrument and a single card that accepted a single sample to do these assays, to a high throughput situation where we would accept samples out of well plates and place them into a device that could accept multiple samples. And again, do multiplex assays in either for proteins or nucleic acids.
Where did the name Codetta come from?
The Codetta was suggested by Hamp Henley, and which means a little tail in Latin, and our devices have little tails. The features that we do assays in have sort of a tail feature, so that fit well. And we did end up adding bio to the name Codetta Bio, that we needed that to make sure we weren't conflicted with other company names that were out there.
What were some of your goals for starting Codetta Bio?
In spinning out companies from my lab, I have specific goals. One of those is actually taking federally funded research and actually delivering products out of that research, having an impact on society. So that's primary goal. My research group over the years have been a mix of staff scientists, postdocs, and students. And in an academic setting, staff scientists, they are sort of in precarious positions, supported by grants, and when grants go away, they may no longer have a job. So one of my goals has also been to start companies so these staff scientists and engineers have an employment opportunity, a place to go, which I think startup companies is one of the more exciting places for scientists and engineers to land these days. I've been at national labs, academia, and associated with startup companies, and I think the startup companies have been some of the most exciting times in my career. In the case of Codetta, I also wanted to have it placed in RTP. It wasn't a requirement, but a desire. It also made sense because at the last several years at UNC, I was running my research group entirely with staff scientists and engineers, and so they tended to be older sorts of people with families and spouses, and so it was more challenging for them to move than, say, a graduating PhD student. So it made a lot of sense to locate the company here in RTP, and obviously makes it easier for me to be involved on a closer day-to-day sort of basis with the company.
Can you tell us about the group at UNC that developed Codetta's technology?
Yeah, so I feel like successes I've had in research over my career has largely been my ability to pull teams of people together, multidisciplinary types of teams, scientists, chemists, physicists, engineers, biologists, and develop teams of people who work well together. And certainly that occurred in the case of the team that was funded by our DARPA efforts in the early days. Hamp Henley was certainly a key member of that team at UNC, and now at Codetta. I ended up running sort of 4 different subgroups in my UNC activities, and had a staff scientist in charge of each of those subgroups, and Hamp was in charge of the DARPA program that developed the Codetta technology and IP.
To find out more about Codetta Bio and its technology, visit us at codettabio.com.